thing is I would request the backbenchers to just come to the front, because today I don't want to use the laptop, because you have already got this lecture from Dr. Talat, I suppose, humoral immunity. So it will be better to explain it on the whiteboard. So whatever are the problems, we can discuss them. I'll just read out the questions that were sent to me. That what is the difference between T-dependent and T-independent response? How B cell is activated and mutated? What is the difference between plasma cell and memory cell? What is clonal selection? What is the process of somatic hypermutation? What is the difference between primary and secondary immune response? Are these all the questions, or there is something else that you would like me to discuss? So I'll start, as a stepwise thing, as a story, and we'll see all these things answered by the, end of the session. If something is missing, you can just remind me. Is that the reason why it is kept here? Okay, just try and fix it. By the way, why there is a chair here always?
Because of Islamic culture.
He sits here. Okay. So, we'll start with so humoral immunity is all about B cells and antibodies. There are the word humoral means something which is soluble in the blood, and one of the things that is soluble in the blood is antibodies. But there are other components as well. So humoral immunity alone does not mean just B cell and antibodies. It's the major part. The complement proteins, there are cytokines, there are other chemical mediators, C-reactive protein. They are also humoral components. But in general, when you will talk about immunity, somebody says humoral immunity, it's about B cells and antibodies in general. Then there are two main types of B cells. One is called as B1 cell, and another group is called as B2. This is about 90 to 95%, while B1 cells are very limited. This is the conventional or the main classical B cell. Now B1 cell, what is it about? B1 cells have got, so all B cells have got one receptor, and that is called as CD19. So all B cells have got one receptor, CD19. But there is one receptor which is very different between B1 and B2, and that is CD5. Very good. While the conventional B cells, they have got immunoglobulin on their surface. So there are two types of immunoglobulin that are present on the B cell surface. Which of those you can remember? IgM and IgD. Now based on these two, the conventional B cell is again divided into two types. And what are those two? One are called as follicular B cells, okay, and one are called as marginal zone B cell. Now this is with respect to which secondary lymphoid organ and which location they are present. So normally follicular B cells will be present in any follicle of the lymph node, of the spleen, of Galt. That is, do you know what is Galt? Anyone? So lymph nodes are the recognized, station houses of immune response. But there are cases where you do not lymph nodes, just the aggregates of lymphocytes. So if these aggregates are in the mucosal tissue, they are called as MALT, malt. If they are in the GI tract, they are called as GALT, galt. Okay, so or in the respiratory tract they are called as BALT. So any lymph node or lymphoid like structure, they will have rounded aggregation of B cells, and that is called a follicle. So there are follicles in the lymph nodes, there are follicles in the spleen. So wherever are follicles, B cells will be there, especially the one which has got IgM and IgD. So these are called as follicular cells. Then there is a special zone in the spleen that is called as marginal zone. So around the margin of the follicle, and these cells, marginal zone B cells, are in just that area. What is different between these two? First, the location, and second is they have got just CD19 and IgM. IgD is missing. So this is the difference. They are CD19, CD5 positive, B1 cells. B2 are two types, follicular and marginal zone. If they have got both the receptors and they are present in the follicles, they are called as follicular cells. If they just have got IgM and CD19, they are called as marginal zone. So on the base of receptor and on the basis of location. Now coming to their function. So functionally, all the humoral immunity, antibody B cells will produce plasma, or they will differentiate into plasma cell. Plasma cells will produce antibodies, and those antibodies are going to react against extracellular pathogens. Okay, so this is the general phenomena. Now, if the pathogen is made up of protein, then dendritic cell will take it. So all the antigen presenting cells will take it up, chop it down, prepare the peptide and present it to T cell, and the B cells will, because B cells are also one of the antigen presenting cell, so they will present that peptide and need help of the T cell. So whenever there is need of T cell help, such a response is called as T dependent. And when there is no need of T cell help, what are those situations? When your pathogen is made up of polysaccharides, nucleic acids, or carbohydrates, lipids. These things, they do not require T cell help. So such responses will be called as T independent. B1 cell, they are T independent. So simply TI or T independent. B1 follicular cells, they are T dependent. And the marginal zone, again they are T independent. So simply here you need T cell help, and we will see what is this T cell help. So this follicular cell is T dependent, it needs T cell help, and what is the peptide or the antigen? It is always the protein. While in case of this and this, it is going to be, one example is LPS, carbohydrates, and nucleic acid. So these are the examples of which antigen will be recognized by this and this cell, and the folliculars are T dependent. They are always going to deal with the protein antigen. Now when this B cells, they are, okay, another thing, what is the source? B1 cells are produced in all the spaces. There is a pleural space. You know what is pleural space? Space around the lungs. Okay, there is a pericardial space around the heart. So and the peritoneum, all the spaces, these B1 cells are mainly produced in the spaces, and they are going to regenerate by themselves. So they are not produced in the bone marrow, they are produced in the spaces, pleural space, pericardial space, or peritoneal space. So they are produced in the spaces. Then another thing is, this group and this marginal zone and B1 cells, they do not have memory. Okay, so the plasma cells have very short half-life, and the antibodies will also stay not too long. So there is no memory here, there is no memory here. Memory is just in T-dependent responses. So we will write down here, it is just characteristic of this group? Main group, follicular B cells. No memory here, no memory here. Mainly, the antibodies that are produced here is just IgM. The main antibody here produced is IgM. But the antibodies produced here are IgM, IgA, IgG, and IgE. Okay? So far everything is okay? Okay, so I have written the immunoglobulins that are secreted in the blood, which you can measure. The IgD is also produced by these follicular cells, but they stay on the surface of the cell. They are not secreted, you cannot measure them. That's why there is no point of writing IgD here. So you will not require class switching because they will be released into the blood. And with context of the infections, allergies, I will discuss this part later in the end. So far everything is fine. Now, one aspect, when the B cells are produced, they are produced in the bone marrow, and if you remember, I showed you certain gene segments, variable segment V, J, D, and constant region, and then there was a mixing of these gene segments to produce millions of B cells. When this is happening, it occurs in the bone marrow, and there is a word, because I'm telling you all this to just confirm one word that is called as somatic recombination. Do you remember this word? So in the bone marrow, when the B cells are getting matured, so this is immature stage, they then finally come out as mature B cells with IgM and IgD and CD19. During this phase, there is something which is called as somatic recombination. Your primary immunodeficiency lecture is done, isn't it? Did they talk about RAG1, RAG2 deficiencies? No. Yes? Maybe. Okay, anyway, here is the enzyme, just RAG1, RAG2. These are the two enzyme recombination activating gene. So the B cells, you want millions of B cell types. Okay, these millions of B cells are produced in the bone marrow by the process of somatic recombination and under the influence of two genes, RAG1 and RAG2. So now we'll come to another aspect, and you will understand why I talked about this, because sometimes it creates little confusion with something else. Now I'll draw a big lymph node or a spleen so that we can see what is happening actually. Why I am making it lymph node or a spleen? Because if a pathogen is entering any tissue, your hands, your feet, any other tissue, it will be taken up by the dendritic cell and taken to the regional lymph nodes. But if a pathogen directly enters the blood, it will be taken straight to the spleen. So blood-borne pathogens go to the spleen, tissue-derived pathogens go to the lymph nodes. Now that's why we just, for the sake of simplicity, lymph node or spleen structure. Then there is a big structure inside the lymph node that is called as a follicle. So I will just draw one of them. With three zones. One is the darker zone, one is the basal lighter zone, then there is an apical lighter zone. This is a follicle, and this inside is called as the germinal center. This part is the cortex, this is the medulla, and from here the hilum, the afferent vessels or efferent vessels, they are around the lymph node. Now what we want to see is what will happen if a bacteria or a pathogen enters your body. So first thing is, if let's say it entered into your skin, the bacteria will be first captured by the dendritic cell, isn't it? And it will be recognized by the help of which receptor? PRRs will recognize it. So we will draw that So the dendritic cell captured it and brought it to the lymph node, inside the lymph node. It was recognized, and then with the help of MHC class 2, it will be presented to T cells CD4. And what are the co-stimulatory signals? Yeah, B7, CD80, 86, okay, or CD28, 80 and 86. These are the co-stimulatory signals, and this T cell is, so normally each T cell or each B cell, they recognize just one antigen. Let's assume this bacteria is recognized by this, among all the millions of T cells, this one just recognizes it. So once it's recognized, this will be primed, so it is watching it for the first time. Okay, so priming took place, and I will tell you this part, paracortical region is actually this, but just for the sake of drawing, this is in the paracortical region, priming has occurred. On the other hand, here is B cell. B cell has got CD19 and IgM, IgD on its surface. This also recognizes the same bacteria. So out of millions of B cells, only one B cell which can recognize this bacteria, they have, interacted with each other, and B cell has been activated. So this is now active. Now one of your question was that what is clonal selection? So out of those millions of B cells which are circulating in the blood, and out of millions of T cells that are circulating in the blood, for simplicity, one T cell recognizes this bacteria and one B cell recognizes the same bacteria. It's the same bacteria. When this B cell is activated or primed, or this T cell is activated or primed, this is called as clonal selection. That one clone has been selected which is able to now produce lot of B cells or lot of T cells against this same pathogen. Yes?
What activated the B cell?
Direct this BCR, B cell receptor binding. So IgM, IgD, CD19 will bind to the antigens on the bacteria. Plus, it will also be helped by the complement proteins, because they bind to it, and then there are complement receptors on B cells.
The bacteria come to the follicle?
Yes, it will circulate to the follicle. Okay, so here it is bound, this is activated, and some number of B cells will be produced now. So now you have got certain number of B cells. This is clonal proliferation. They have multiplied, and once they are activated, certain changes takes place in the receptors of the B cell, and these B cells will come out of the germinal center into the paracortical region. Now I will write here is activated. So this activated B cell which has been multiplied against this bacteria has now come out of the germinal center, and now it will interact with the activated or primed T cell. Okay, at this stage, now this activation, B cells have got MHC class 2 expression or not? Yes, because it's a antigen presenting cell. So it will now chop down this Another thing is, when these antibodies bind with the bacteria, they take it inside, internalize the bacteria, break it down into pieces, load it on MHC class 2, and go out of the germinal center and present it to T cells. So here now there is the MHC with bacteria on its surface. So MHC class 2, here is of course CD4, and now the co-stimulatory signal will be what? No, it's CD40 and CD40 ligand. So here is CD40 receptor, and here is CD40 ligand. And one special thing will take place, that now the word T dependent or T cell help Here is the help. This T cell will now ask this B cell, or changes this B cell into which type of antibody to produce. So now I'll just take you on the side. We will see 3-4 other things, that this dendritic cell, based on the antigen, will decide whether this T cell has to become a Th1, a Th2, or a Th17 group. If dendritic cell produce IL-12, it will become Th1. If it produced IL-4, Th2. If IL-17 and TGF-beta, then it is Th17. Now we'll link it with the infections now, that if it's an intracellular bacteria, Th1 cells are capable of producing IgG immunoglobulins. We will not talk about the different types, IgG 1, 2, 3, 4, just simplicity. Th2 can produce IgE. Th17 can produce IgA. Okay, so this is against parasites. This is against viruses at mucosal surface. And this is against bacteria. So how it will happen? At this level, when the T cell will give help to B cell, this T cell will decide. If it is a Th1, it will cause these B cell to produce IgG. If it is a Th2, it will cause B cell to class switch to IgE. If it is a Th17 at the mucosal surfaces, then it will ask the B cell to start producing IgA. So far everything is fine. Now once this has occurred, this activation has occurred, these B cell, T cell, this combination, it's going to move back into the germinal center. This combination, this together, B cell with T cell, will move into the germinal center, and now the multiplication of the B cell is very rapid. They can multiply very, very fast. That's why what will happen, this B cell number too much increase here now. And because of lot of B cells, this zone is called as dark zone. And this combination, where is it now? This combination comes here. T cell along with B cell, and there is another new cell here, which is follicular dendritic cell, FDCs. Follicular dendritic cells are very different from all the dendritic cells in the body. They just stay inside the follicle and nowhere else. They do not have MHC class 2 or MHC class 1 expression. They only present the antigen to B cells. Sorry, it's a wrong. B cell is here, T cell is here. And these T cells, when they move into the follicle, now they are called as follicular helper T cell. The T cells that were primed by the dendritic cell interacted with the B cell. Together they moved into the germinal center. Now it is called as follicular helper T cell. This is follicular dendritic cell. Both of them, along with the activated B cell, are now present in the germinal center. Now here another new term. So this B cell has recognized the bacteria, the same bacteria. It has started producing the antigen binding affinity, which is good enough. They have recognized it. But is it the perfect fit? So I always give this example that when you make a key, you go to the key maker, you give him your key, and then he first make the as closest match as possible in the machine. And once it is done, sometimes still it is not opening because it's not the perfect match. So he has to do little more tweaking around, make it a fine fit. That fine fit is called as affinity maturation for the perfect fit. Okay, so how he do it? He do it by changing it and making it more sharp or whatever. Here B cells, they start multiplying, and there is one special gene here, and its name is AID. Activation induced cytidine deaminase. This enzyme will change the affinity, so each B cell, it will bind to this bacteria with different affinity. One may be bind like this, another little stronger. Some will bind very, very strongly, and that is what B cell want. Very strong binding. This very strong binding is called as affinity maturation. Okay, and these different varieties of binding is produced by activation induced cytidine deaminase, and this process, so the genes will be again cut down, spliced together in different combination by different mutations. That's why this process inside the B cells of the genes is called as somatic hypermutation. Okay, so here it is called as somatic hypermutation. Now, if you see there are two different words. This is somatic recombination, and this one is somatic hypermutation. They are both playing with the genes. So there the genes involved are RAG1 and RAG2. Here the gene involved is, the enzyme involved is AID, activation induced cytidine deaminase. There the purpose is to produce big library of B cells. Here the purpose of this recombination is to have a perfect fit. So now do you understand the difference between somatic recombination and somatic hypermutation? Yes, please.
The dendritic cell binds B cell.
So now you are talking about follicular dendritic cell. Yes, we'll go to now the next stage. So first I want to explain this, then why B cell will bind and what it will do. Yes.
Is it the same as affinity maturation?
So when this somatic hypermutation occurs between the genes, it will result in affinity maturation. This is how the cells will mature. So now at this stage, follicular dendritic cell, it has also found this bacteria, okay, with the help of Fc receptors, and it is going to present this again now to B cell. Okay, which B cell? These B cells who have resulted in somatic hypermutation, one by one they will go inside and try to check the fit. So if the fit is weak, it's not very strong, this helper T cell, follicular helper T cell, he is not happy. So when he is not happy, he does not give the survival signal to the B cell. So that's the relationship. The job of follicular dendritic cell is to present the bacteria again to the B cell and check if the fit is good or not. If it is weak fit, T cell will not give a good signal, and this B cell will be deleted, it will be destroyed. So that's why in the dark zone there are a lot of B cells. One by one they are going up. If they are not selected, they are deleted. That's why in the lighter zone less cells, in the apical zone even more lighter. So that's the reason. They are big number, one by one checking their fit, bad fit, cancel, deleted, another come. So finally what you have now, some of the very good, nice fits, the best fits. Now these B cells are called as, they are affinity matured. Okay, does it make sense that hypermutation was changed in the genes to find out different fits? All these fits of these B cells are different. Follicular dendritic cell will check the fit. When the T cell is happy, what T cell does? They produce something which is called as BCL2. Excellent. So BCL2 is the survival signal. The survival signal will result in best fit B cells, and they are affinity matured now. And now they are ready to leave the germinal center, go into the circulation, convert into plasma cells, and those plasma cells will produce either IgG, IgE or IgA depending on the initial signal. Yes.
What about memory?
So once the job is done, so they go out, now they are able to produce best fit antibodies, okay? They will fight the pathogen. How they will fight? Let's say IgE, it will bind to the parasite, eosinophils will come, degranulate, destroy. When this is done, out of these, let's say we have 200 cells, those 200 cells will die, only 2